Here we go. On this slide, the only thing I asked you to note is the microvilli, which might be a little bit new from the cell organelles you learned in Biology 1. These are little extensions of the cytoplasm. I think of them as fingers. And the purpose of these fingers, as we discussed, was to increase the surface area present in a cell, which allows for increased functionality. And the example we gave was of a digestive cell, which, given more surface area, will be able to absorb more nutrients into your body. The plasma or cell membrane was the one cell part we spent, I guess, all of our time on. I don't know if these prefixes will be used on the quiz, but you should probably know them. Intracellular fluid and extra or outside of the cell fluid. The glycocalyx functions in cell recognition. We talked about good guys versus bad guys. The glycocalyx is made of two different kinds of molecules, glycoproteins and glycolipids. Recall that glyco refers to a carbohydrate component, and protein, a protein component, and lipid, a lipid component. The fluid mosaic model, that term was used. The cell membrane, remember, is a bilayer of phospholipids. There's also cholesterol in the membrane. These glycolipids, glycoproteins, and regular old, what we called, I guess, purple proteins jutting through the membrane there. The primary component was the phospholipid. Again, recall the dual nature of your phospholipid molecule. A polar head, which is the phosphate group, nonpolar tails, which are the fatty acids. Those nonpolar tails try to keep themselves away from the water, so they back themselves up to the interior of the membrane. That's hydrophobic. And the polar head was hydrophilic. It is attracted to water and thus faces the water on the outside and on the interior of the membrane. Remember, proteins, the integral proteins come in two varieties. Those that have channels in them are used in passive transport. Those that do not have channels in them, which are usually used in active transport. Cholesterol is going to not look like a protein, not look like a phospholipid. It's going to have a different structure, and the cholesterol adds fluidity, or kind of a more liquidy nature, to the cell membrane. The glycocalyx will be anything jutting out of the surface on the extracellular side. We then spent some time on the three types of passive transport. Remember, anytime we say passive transport, we're implying there's no energy supplied, meaning you're going with the gradient, or according to nature, or gravity was the analogy. So from where it's more concentrated to where it's less concentrated. In simple diffusion, we're going directly through the bilayer, through the phospholipids, and those were nonpolar lipid-like things. Again, we listed many examples of those. If you need help because you are polar, which is not mentioned here, we'll add this. So if you are polar and you need help, all these things have polar, have charges on one end or the other of the molecule, and you're going to need the help of our protein channels. Again, these are usually these proteins in the membrane with a channel in the middle of them that allow these ions and sugar molecules. So if we, again, magnesium was an example of an ion I used to skate through the membrane without coming in contact with the phospholipid bilayer. And then water has its own special category. Don't call water simple diffusion. Don't call it facilitated. We call water, the movement of water, osmosis. And on the next, we see how, again, that exception of water, how water can move directly through the membrane, as in simple diffusion, or it moves, since it's polar, with the help of a protein. So that's why we give water his own little name here, osmosis. Both these pictures here in the center refer to facilitated diffusion. You have molecules such as sugars and ions which need the help of the protein to move through the membrane. And then over here on the far left you have simple diffusion for lipid-like materials passing directly through the lipid bilayer. In terms of osmosis effect, we dealt primarily with semi-permeable membranes that allowed the water to move across, but not the solute. This creates the movement of water in one direction or the other and creates that swelling or shrinking of the cell. So in this example over here, we note that water has moved from left to right. And the way we establish that is we note the solute concentration. The solute concentration is higher on the right, which means there's less water here. Less water, right, because the water is opposite the solute. There's less solute on the left, which means more water. And water follows its own gradient from more to less. These were terms you have to know. Isotonic is self-explanatory, but mainly you have to distinguish hypertonic, which will cause a cell to shrink or crenate, and hypotonic, which will cause a cell to swell and potentially burst or lyse. These terms refer to the hyper, as in hyperactive or a lot, refer to the solution. Hypo meaning low, again refers to the solution. So we do hypertonic first. A hypertonic solution is high in solute in the solution, in the environment, in the beaker, in the aquarium, wherever the cell finds itself. And the inside would have less. Again, the solute concentration is opposite of the water. So a high concentration of solute means a low concentration of water. We'll go blue here, L for low water. A low concentration of solute means a high concentration of water. Water goes from high to low or more to less, and this is osmosis. This cell would shrivel or shrink or crenate. In a hypotonic environment, hypo is low. Again, we're talking about the solution relative to the inside of the cell. Again, recall that the solute concentration is opposite that of the water. So a low solute concentration means a high concentration of water. A high solute concentration indicates a low concentration of water. Water goes from high to low, and this cell, like a hippo, will swell. And then opposite of passive transport, you have active transport, in which you're moving things from a lesser concentration to a more crowded, a more concentrated area. This is uphill, so to speak. This requires ATP, which is your body's form of energy, cellular energy. And the one example we talked about was an antiport system, the sodium-potassium pump, in which these are moved in opposite directions. The sodium-potassium pump, 3, 1, 2, 3. And again, we did this in class, the pump. 3 sodium atoms are pumped out of the cell, and 1, 2 potassium ions are brought into the cell. This differential exchange of 3 positives for 2 causes the inside of the cell to be negative relative to the outside. This pump is moving these ions against the gradient, uphill, so to speak. In the center here, you have the indication of the gradient. So sodium wants to move in. The reason for that is because the sodium is in a higher concentration outside of the cell and in a lesser concentration inside the cell. Potassium wants to move out because potassium is in a higher concentration inside the cell and a lesser concentration outside the cell. But even though potassium, the gradient is pressuring the potassium to move out, we're going to pump it in, And for sodium, even though the concentration gradient pressure is to have the sodium move into the cell, we're going to pump it out, which is why we need ATP, or energy. Creating this negative charge inside the cell is called the resting membrane potential. It's a way to tell the cell that it's in at rest, or it's in the off state. We mentioned like a light switch. Sometimes when you're moving substances, it's not the concentration that's the most important thing, but it's the size of the particle. Certain things that are extremely large are going to require energy and ATP regardless of concentration. If you're moving the substance out of the cell, we talked about neurotransmitters and their relationship to recreational drugs and how they affect the brain. These are pumped out of a nerve cell via exocytosis and endocytosis, like a white blood cell, we said, swallowing up a bacteria. This picture gives you an example of exocytosis, how the membrane of what's called a vesicle fuses with your main phospholipid membrane, and the particle is excreted, or this would be the opposite of that. Here you have a cell extending its cell membrane to swallow up a bacteria, otherwise also known as, we mentioned, these charts. The one other thing you should know is some example. Given a specific molecule, you should be able to tell me what kind of movement it uses to go through the membrane. So for simple diffusion, O2, CO2, H2, any diatomic molecule, along with fats, steroids, lipids, fatty acids, anything that indicates that you would be sure would be nonpolar. Facilitated diffusion is for glucose, ions, anything with a charge other than sodium and potassium, and amino acids. These we know are polar. Osmosis is for water. Active transport according to the pump is for sodium and potassium. And active transport by exo or endocytosis is for large particles. And I'm not going to quiz you on pinocytosis. Not too bad. Good luck.